So first of all, thank you very much for the kind introduction and to the whole KNIME team for the invitation to give us the opportunity to tell you about our work. Most of you will probably know Bayer. It's a large size pharmaceutical company, and as such, we are interested, of course, in doing drug applications and bringing drugs to patients in order to help patients. So we are seeing here now the focus on oncology, obviously. So in order to bring drugs to the patients, pharmaceutical companies need to submit data to regulatory authorities such as FDA or EMA, and those authorities require companies to submit data in certain standards. So therefore, pharma companies or CROs, which are clinical research organizations, create study results in a programmatic way. And those data sets are usually referred to as TLFs. So that stands for tables, listings, and figures, which actually contain the study data and the study results. Usually a study consists of multiple derivation levels, and the usual flow is depicted here. So we have the eCRF or EDC, that stands for electronic case report form or electronic data capture, that happens at hospitals or sites where the study is conducted. So investigators literally type in data into report forms. So the first data that arrives at CROs or clinical companies or pharmaceutical companies are raw data. And then certain standards, STTM, which stands for Study Data Tabulation Model, and ADAM data analysis data models are required by authorities. And once those data set exists, finally the CSR is created, that is the clinical study report, and it's sent to submission to those authorities. And this process is much more complex, as each data stage has several iterations of data cleaning steps, and multiple additional standards and guidelines need to be considered and to be in place. The standards are defined mainly by CDISC, that is the Clinical Data Interchange Standards Consortium, that is a standards developing organization who defines those standards. And you see here an excerpt of the CDISC website, and behind each of those dots are guidelines, and a guideline can be a multiple hundred pages document that specifies how a certain data set should be implemented or how certain standards should be implemented. So now what are ADAM data sets specifically, and what are domains? So both the STTM format and the ADAM format is highly interconnected, so you can not have one without the other, you could say. And so the STTM domains are depicted here, or some of them, and a domain is actually a collection of logically related observations. So for example, a very common standard domain is the demographics domain. So you can think of it like a huge table that lists all the subjects that are enrolled in the study, and where the demographic information of those patients are listed, like sex, ethnicity, race, and so on and so on. And not all domains are necessarily need to be present in a certain study. So indication specific and also study design specific, there are differences, but some of the domains are usually present in each study. Some background how this clinical program is now actually conducted. So SAS is a commercial programming language, and it is the de facto standard in the pharmaceutical industry and for regulatory agencies. And usually the TLFs are programmed by SAS experts within CROs or pharmaceutical companies. And the implementation of the mentioned CDISC standards or other standards are specified company-specific by SOPs. So how the standard should be implemented, how certain processes are implemented, and so on. And considering all this highly regulated processes, that leads to highly interconnected dependencies that actually makes it very difficult to try to deviate from that approach. So furthermore, not all functions that are involved in those processes are necessarily trained programmers. So for example, data management teams who should take care of cleaning raw data or other derivation steps of the data are not necessarily SAS programmers. And although we speak a lot about standardization, each study is a little bit different. So maybe 80% is standardized, maybe more, maybe less, but there needs to be a considerable amount of adoption from study to study. And that is especially true for efficacy domains, for different endpoints that are defined by more or less complex study designs. That might be especially true in oncology, where study designs are inherently complex. And that, of course, is a challenge because it costs a lot of time and effort to adopt from study to study. And furthermore, this industry dependency for certain software, as it was in the past, does not allow us a realistic chance to use KNIME for submission to authorities. So that's a clear disclaimer. Currently, it's more used or considered by us as a case study or a feasibility study to prove that other technological implementations are possible. And we want to spark ideas. And we would like also to establish KNIME, as it was mentioned earlier, as a Swiss Army knife for non-regulated internal work with clinical data. And just to clarify, neither CDISC nor the FDA nor the EMA does say you have to use SAS for submissions. You are free to use other programming languages, but as I said, since the industry is so much adopted to SAS, it's very difficult to step out of that. So why we think wirral programming could be interesting in this domain? So usually the workflows and the analysis we conduct need to be validated. So that means usually they are double programmed. So that means that two clinical programmers create a TLF independently, and then it is compared whether or not they reach the same result. We believe that a double programming alternative could be KNIME. What is especially interesting here is that the visualization of the data flows are actually the documentation. So you can look at it, you can understand it very easily. It's very different from a programmed script where it is not inherently clear what is happening. So, and therefore you can also not discuss workflows. You can probably have a harder time to learn it. And we believe that KNIME could serve as the lowest common denominator between, let's say, data science affiliated functions and programmers. And the immediate clarity of the algorithm or of the workflow also allows to optimize and to discuss workflows, and also invites for experimentation, as it is very easy to branch off at a certain step of a workflow or derivation step. Yeah. So maybe just to clarify what we are doing or how the data looks like and what are especially suited for the clinical use here. KNIME comes with a native SAS 7B.DAT reader file, which is very nice. So that is obviously the proprietary data format, which comes out of the usual SAS scripting language. So again, KNIME can read that natively. We can also very easily extend such a workflow to read from remote servers. So that is very nice. And to show you that it's actually very simple ETL, what we are discussing here, we have here this little example where we just use the column expressions node to do some data grouping. So we have here a numeric value, and we need just to group that into certain buckets. I mean, again, that is what CDISC standards look like. Yeah, so the requirement to have this variable in this derivated step here, yeah, it is required. So and other commonly required ETL functionality, you probably all use those nodes: concatenate, column appenders, rule-based row filters. So no rocket science what we are doing here. We will come later to what where KNIME really can shine in this context. So we have here the example of an ADSL derivation. So ADSL stands for Analysis Data Set for Subject Listings. That is actually the coming out mainly of the STTM demographics domain. And so we separated this workflow, of course, or structured that into the data retrieval part. I will show you in a second what we used for this demo workflow. We have a derivation step, and we have a testing and XPT output. I will also show you in a second what that looks like. So we collected this mock data, which we presented at FUSE from the FUSE Open Data Repository. So that is a publicly available data repository that provides clinical mock data sets for testing purposes. So we used here this Postgres SQL connector. We have a very simple SQL query here reading the data set. So here you have one example how that looks like. And maybe to clarify about the size, so you see here it's not a big column, right? We have here 306 patients and 25 columns. So this is how we retrieved our data set. So, and I will show you now just very few steps out of this workflow, which might be a little bit more interesting. So most often when doing data derivation steps with clinical data, we cope with date values. And as you all might know, date values can be a real pain. But here the KNIME date nodes or date and time nodes come very handy. So we read a data set, and we have a thus date representation, which is an integer that defines the day, the days that are passed since the defined thus date origin, which is defined as the 1st of January 1960. And we can simply define those origins. KNIME does then this shift to actual dates, KNIME dates, and then we can do very nice calculations and calculate, for example, here some durations since the treatment start date of the patients in days. Again, a required CDISC variable. Another example we worked on is the ADTTE domain, that is the time to event domain. And there I want to give a little background information. So time to event usually encompasses survival analysis. So what is survival analysis? It's a very general method it's statistics that is designed for modeling time to event data. And that is used in very different fields like sociology, economics, engineering, and concrete examples could be a failure in mechanical systems. So when does a machine breaks, or in our clinical context, it's the survival of a population past a certain time. So to be more precise, in the oncology context, it is the time to disease remission, progression, or death for cohorts of patients, or the subsequent comparison between different treatments within a clinical trial. And the events, what is special about this clinical analysis is that events typically are subject to censoring. So that means we have a lot of missing or incomplete information for a variety of reasons. So for example, subjects are lost to follow-up. So we simply do not have any follow-up information about a patient. That happens frequently, or the patient drops out of a study for certain reasons. So for example, he decides to do another study, uses another medication, or has a serious adverse event. And those which subsequently leads to a dropout of the study. And this influences the statistics, of course, quite significantly as the power decreases by censoring. Because likely, if you have less subjects, the uncertainty increases. And we are usually interested in the survival function and the hazard rate. And by subsequent tests, we can differentiate between different treatments with using the null hypothesis that groups have the same hazard or are not different. And what is very nice is that KNIME comes with a node for that, that already allows us to do half of the required work here. So we can simply encode certain variables by Boolean factors and then pass it to the Kaplan-Meier estimator node, which calculates the statistics we require and also come up with this nice survival plot. So here you see those crosses means censored patients. And this is what you usually will also see in a lot of clinical studies. This is actually what is one of the primary endpoints usually you are interested in. And but just a word of caution, so the details matter. Usually there are subtle changes in different programming languages with respect to certain statistics. So for example, confidence interval calculation differs, for example, between SAS and R, and there is also with KNIME, and we also have different roundings probably. So just to take care of that. So finally, if we want to write the output, we do that in an XPT format that is in SAS inter-exchange format. So SAS can write this and read this, but usually, as you saw earlier, SAS also has a proprietary format. But this is an output format that the authorities accept. And although KNIME does not provide a native XPT writer node, just a hint to KNIME, probably provide one, we would be very happy. But it isn't so important because we can also help ourselves here with using an R snippet node and make use of the Haven package that actually allows us to write this output format. So this R snippet actually just contains this one line. It writes this file format for us, and that would be theoretically ready to ship to authorities. So now a very crucial part of the whole clinical data analysis is GXP, which stands for good clinical practice, good computational practice, good like everything practice. And a large part of that is validation, testing, logging, and reporting. So the authorities want CROs and pharma companies to be able to prove that certain tests were applied to a data analysis, that a certain validation took place. They would like to be able to trace data points throughout the whole analysis pipeline I showed you earlier in the beginning, so the whole data derivation process. And we tested what KNIME has to offer here. Because that is usually the first question you are asked if you consider if you demonstrate a new technology within a for statistical programmers or clinical programmers, they will immediately ask, is it validated or can you validate it? And this is why we took some emphasis here. So what is very useful is the KNIME table difference finder, which allows you to find value differences. So for example, we literally found a typo here in the PODR repository. So here an R is missing in this event description. You can also find domain differences. So that means for numeric data that if ranges differ, or for nominal values, if different set values are, or the different sets, the sets are differing. You could consider this domain difference as an additional validation layer. You can, of course, also compare numeric values. We see here slight rounding errors we identified. We see here type differences. So we have here an integer, here a double value. And this table difference finder could be already used out of the box for double programming comparisons. Yeah, we could read a data set that is derived from SAS, and we can read a derived data table that is generated in KNIME and do the comparison and see where are the differences. But KNIME comes with a large repository of testing nodes that actually cover almost all relevant scenarios. And yeah. What is really interesting is the logging capabilities of KNIME. So that is also an authority requirement that you can document how an executed script or what an executed script was actually doing to the data. And we use the KNIME workflow summary here. So you can export this very verbose XML document that actually traces everything that happens within an executed workflow. So you can either use third-party tools to read and analyze that XML document, or you can use the KNIME XML reader to read that back into KNIME and use the analyze workflow summary to display what data is within that XML. So information that is provided are the installed plugins, the used nodes, the operating system, when was the workflow executed and by whom it was executed. And what is really interesting in the next step is that each data point is actually traced. So for each node, the input, the state, the configuration settings variables are reported, how the data looked at the input port, how the data looked at the output port. And you see here from this little examples I showed you, 80,000 lines of logs are generated. And that is more verbose than a usual SAS log is. So actually, again, everything is covered here. So to summarize, we believe that KNIME is a feature-complete and powerful alternative for working with clinical data. So we demonstrated that clinical data derivations of certain domains is possible. It could be an alternative approach for double programming or validation of SAS derivations. And it has very verbose logging and validation capabilities. And on top, the visual programming, generally speaking, could be especially useful for pipeline optimization, automation, and of course for educating less tech-savvy or non-programmers, and therefore has a great potential, we believe. On the downside is, of course, we have a highly regulated environment and we have existing standards. Industry adoption is not straightforward, even for other statistical programming languages, which might be even closer to SAS. And the existing pipelines, SOPs, and need to be considered. And of course, study continuity needs to be granted. So that means, there are many studies running, probably since several years, many studies run in parallel, and it's very difficult to say now we change things. From now on, we use KNIME. So that is a very difficult topic to do this this mind shift towards other technologies which clinical programmers are not so much used to. So I would like to thank Seema, Anupama, and Cornelia from Bayer, and Jose from Fuse, who provided us the PODR access. Special thanks to my colleague Clara Beck. Special thanks to my colleague Clara Beck, who co-developed or co-led the development of those workflows. And with that, I would like to thank you for your attention and would ask for questions.
Fantastic. Thank you, Robert, very much for this nice presentation. And special thanks goes to you bringing on a totally different topic, which probably is pretty new to most of the KNIMers or data scientists or people working in the R&D sector of pharmaceuticals. It's not typically what we talk about, and it's not typically what we use KNIME for. But I'm so happy that you did that and you just demonstrated how to use KNIME in this environment too. And very connected to this one goes the very first question. KNIME is not really a known tool to be used in clinical trials. How do you get to use it?
So I have a quite diverse background in Bayer. So I came from very early research, bioinformatics, over information retrieval, subsequently to the clinical development, and I brought it with me. And I saw great potential there as we realized how constrained actually the clinical programming functions are by sticking to certain programming language and certain paradigms they never questioned actually. And we saw a great potential here, and this is why we just said, let's apply that, let's demonstrate that. And it was actually well perceived by our programmers. So they are very interested in, again, the biggest hurdle here is really the existing standards and existing technologies that are just there since decades.
Mm-hmm. Thanks. I think there are like a lot of follow-up questions to that. So let me try to summarize the other one, which is around this question. You already mentioned that SAS is, well, unfortunately, the de facto standard. How far are you with the idea to use KNIME in this domain? Is this something like an official project for Bayer? Well, as much as you can comment on this, of course.
Yeah, no, I mean, it's definitely not what I said in the beginning. I mean, we are, so I have the privilege to work in a very nice innovation department within clinical development. So that is also a quite unusual or modern approach. So therefore we have some freedom to experiment and to spark ideas. And this is actually what we wanted to do here. And it's not realistic that we use KNIME for submissions, or for other external data submissions. So for me personally, KNIME is a great personal productivity tool, and I believe that several functions working with a lot of data, especially with messy data like data management, could benefit from that, but that are internal processes. So, I mean, with demonstrating here the generation of ADAM data sets by considering CDIS standards, we already moved to the end of the possibilities, right? We tried to find the most difficult thing. But I think with that, we proved that KNIME could actually be used for clinical data handling. And there are many opportunities, I believe. And that is what we wanted to demonstrate and spark ideas for.
Yes, and I believe you did a fantastic job on that. Let me just still continue with this one a little bit, because Ken was chipping in with his own question, which is more into the direction that you went on with your answer. It says, very impressive. I too think KNIME is a tailor-made for problems like SAND format conversion at my own company. Well, we heard the questions related to that one. Can you speak to anything you're doing to make the change management part easier for clinical teams that are less familiar with KNIME? How can we be successful in retraining an organization to use tools like KNIME?
That's a very good question. I think that is probably one of the biggest problem, not only KNIME related, also generally speaking. Yeah, I know that other pharma companies are looking into submissions with R, but even they have a lot of difficulties doing the internal change management, although they spend a lot of resources on that. However, I really believe if you show the capabilities of KNIME, and I really believe that it's a full stack data science tool, right? You don't necessarily, so for the most common ETL processing, you don't need to leave KNIME. That combined with the connectivity, I mean, we saw that clinical data sets could be read, should convince people. I agree that it has a maybe steep learning curve for someone who is not exposed to data science at all, because it's a completely different thinking, right? Even for very professional SAS programmers, it might be difficult to do this switch in thinking. Yeah. On the other hand, what was also said earlier, I believe that you can be much faster with KNIME, and you can also adopt and reuse parts of your workflows much easier, which again, I think is a pain if I have to read a huge program script and try to reuse parts, and maybe you have like dependencies you are not aware of, but looking at a visual programming language, dependencies become aware immediately. And yeah, and therefore I think we can convince people by its usability and how fast you can be if you master some of the basics.
Well, unfortunately, our time's getting up a little bit, but that is a very good example. Actually, you sparked a lot of questions. There are lots more actually to ask from you, and it seems that this topic is really interesting and it's a hot topic for the community and the people listening to us. Thank you very much for answering these questions. Thank you much for the presentation, and especially that you took the effort to have a little bit longer presentation, so you could introduce also the domain a little bit to those ones who were not very familiar with that. Thanks a lot for that, and I hope you can stay with us for the next session. But regarding the questions, actually, we of course did not have the time to answer all of the questions you posted, but we are going to be posting it in the KNIME forum. So please follow up on the KNIME forum with these questions. They're going to be all there, and the presenters are going to answer that to their best knowledge. Again, thank you very much, Robert, being here with us and having this very nice talk.